Now it's time for the report from the day one breakouts, and we've got three groups. Stephane, Zaffran, and Heather Etchevers on abnormal development in humans, Jesse Gillis and James Briscoe on regulatory mechanisms, and Ali Erturk and Hiroki Ueda on imaging and spatial transcriptomics. So who's going first?
So we will start, I think.
Great, Stephane. Go for it.
So hi, my colleague Heather Etchevers and myself have led this breakout session on abnormal development in humans. We had a good discussion with participants, and we had up to 40 participants attending this session. So as you know, each organ of the embryo or fetus develops during a specific period in the pregnancy. There are three stages of prenatal development: germinal, embryonic, and fetal period. And disruption during any of these stages can lead to abnormalities. But we know that organs also continue to develop even after birth. Such as the heart, cardiomyocytes, they require maturation to be efficient, for example. And pathology observed at birth or even later in the life can be the consequence of abnormal development. So what is the cause of developmental disorders? So we know that the normal process of development can be disturbed by inherited abnormality from parents, like based on the genetic, epigenetic, and that could be also exacerbated by environmental factors or even by metabolic defect. We also, this perturbation can be erased de novo due to de novo mutation on a chromosome or genes, and that is also exacerbated by environmental factors. And finally, any perturbation due to the environment can lead to abnormal development. So during our breakout session, we divided questions in 4 themes. And the first was, what is our priorities? So we think that during the discussion, we find that the stage to study are important, and all of them, because even if the first trimester it's very important for the development of organ, but you've seen that the organ have a specific stage to develop, and even after the first trimester. So all of these stages are important, but that raised the difficulty to the accessibility difficulty to find a specific stage for each organ. And we need to define also the organ-specific windows, and what is the windows where this organ is susceptible to any perturbations. So we need to promote collaboration with pathologists, obstetricians, genetics, pediatric specialty to implement the collection that we have. Due to the human population diversity, we also need to get a better annotation for the ethnic origins. And is it possible to annotate that and also to get information on the exposure from disease side? And we have to compare that to a normal reference, but what is the reference? That will be discussed later. It is also important to be able to stratify for a better evaluation of population risk. And we also come out with this important question regarding the coordinate in each samples, and that is now possible with imaging such as light sheet microscopy, micro-CT, with a high quality of imaging that allow us to have a superstructure coordinate. So it is also important to better genotype samples, but then that raises the question of the level of this genotyping, which level we want to have this better genotyping. And that is necessary to establish the normalcy of samples, at least for chromosome, of course. And is it possible or feasible to do whole exome sequencing genotyping for each sample? It's a question that we raised. It's possible also to do that when we prepare and conceive well the prospective collection that we have with appropriate ethical consent. And to fill the gap, the temporal gap for human samples, we think that it's important to link the data with the animal or in vitro model that labs are studying presently. And for that, we have to establish a correspondence across developmental stages per organism and per organ. And we need a bridge also to international society to better exchange and establish this correspondence. So I will let Heather to continue.
Thank you. Yeah, it was a fantastic discussion. Let's see if I can share my screen here. Great. So is it on sharing? There we are. So we spent a lot of time talking about priorities because that seemed to represent a lot of interest, but then our division into themes, you'll see that some of the things we discussed in the group bled over from one theme to another. So talking a little bit about the kind of data curation that we might need in studying abnormal human development, many people simultaneously, it was quite nice to see, brought up the necessity to go beyond transcriptomics and really move into the era of multiomics, and in particular annotation of chromatin availability, as well as perhaps imaging the physical placement of the heterochromatin within the nucleus at any given time within particular cell types and making those bridges. Working on making consensus annotations of promoters and enhancers at given times in development. That's why Kat's talk was so excellent, an introduction to this summary of the breakout session. We won't take too long because we want to leave some time for discussion. There was a really interesting point that was brought up a little too quickly because we hardly had enough time to talk, but that it's now possible with the mosaicism that we know is present in all the cells of the human embryo at pretty much any given time in its development, depending on the level of sequencing that we do, to do mosaic analyses that might make it possible to conduct studies related to cell lineages within the human embryo. Another thing we talked a little bit about as far as data was concerned is that we need to, well, this gets back a little bit to making links to professional societies, but human genetics has a lot to teach us, and we have a lot to teach human geneticists. And so one way or the other, it's important when we do these sample collections, be they from normal or abnormal individuals, to keep a sample of DNA so that we can perhaps be able to interpret clinical findings later on relative to these references. Another major theme was about the obstacles that we're going to obviously encounter when talking about studying abnormal development. In particular, there was a really interesting discussion, I think, about the difficulty there is about talking about death in the pre and early postnatal years, and with families. You know, some deaths can be anticipated, and we can provide correct consent, or at least we can undertake the process that this consent would be appropriately obtained. But many families don't want to talk about it afterward, and that's perfectly within their rights. So that's a real obstacle. There is a possibility, though, that we can anticipate such obstacles and nonetheless still obtain good samples for this kind of research. And I've summarized a few of the points here. And so what was very interesting too was to hear the perspectives from different places around the world, because there were different possibilities in Europe and in Asia and in Australia and in South Africa. So that was a very, very interesting discussion. Some other obstacles are a little bit more technical. It's just mapping, right? And this gets back to a little what we had considered a priority, that it is important to say, well, what is equivalent to what in our animal models so that we can use our animal models better to fill in the gaps. There was this discussion that has come up in the more general assembly at the HCA, of course, which is about, how do you ensure quality of the samples that we're going to be considering, either normal or not normal? Are they really representative? And that's not an easy question, but we'll take what we can get, and particularly because it's hard to come by these samples. And it was mentioned that it's possible to validate some of these things using the expertise of pathologists. And this will lead right into the idea of setting up references, which was discussed a lot as well. How do we figure out what normal means during human development? What is healthy? So we have to define that. And to be able to talk using the same words, we need to set up these ontologies so that we really do say what we mean when we talk about an endothelial cell in a particular organ. Is it a long endothelial cell? Is it a tip cell of the lung endothelial cell, or is it defined by its position? What are we going to use to talk about our cell types? And we also will need to not only make them organ specific, but also have the possibility of taking a step back. Some cell types may be exactly the same within different organs, and we can do that by putting them all together. All right, so I'm running a little over here, but comparing, of course, what is provisionally considered healthy with what we know is a disease that we want to study will allow us to tease out some of these questions as well. There's a little comment on the last slide, which I think is really relevant here. We did want to bring up this idea that all of these provisional reference systems that we're going to use, or samples, are going to have to be versioned in some way, right? So today, a sample that we consider normal, we might find out that it is not so much so as we thought, or at least not for a particular organ. And so it might still be useful for studying kidney development, not so much so for brain development. And so again, we just have to keep a lot of this metadata available. Accessing these tissues is easier in some places than others. And so that implies that we need to figure out a way to support the international resources that are able to obtain and distribute them and or the data that is obtained from them. And I think this is the last slide. So this gets back to the question that was brought up about multi-omics. That is going to help us define what is normal, right? So all of these annotations are going to bootstrap our definitions. I think we had a bit of a discussion about how, of course, in the first trimester, we're going to see representation of anomalies that never make it to birth, that are not viable usually, or, exactly, they're not viable. This is the comment that I remembered from the discussion. Not everyone is wild type. In fact, is anybody wild type? I don't know. But in any case, we have to keep it in mind, and we always have to have that sort of provisional sense of what is normal. Of course, representation in the human population, this is a theme that comes up over and over again in the HCA. We have to find the ways that we can represent humans overall as best as we can, and that we need to be a little bit careful about the terminology we use. So there's a couple of recent opinion pieces that are cited here. So I'm going to stop here and open up the floor for people who had been in the other breakout sessions to be able to participate in the discussion. Thank you.
Great, Heather and Stephane, thank you so much. And that you guys you greatly, wonderfully exceeded our hopes for what some of the discussion points that you could raise. And so one, the idea of mutant allele effects per cell type, seeing causal mutation effects in a lineage in an organ at a time in space, then, and the idea of de novo mutations, that is absolutely spot on. And I'm wondering, in calibrating that and understanding human variation of levels of de novo mutation, do we need to have parental DNAs so that we have at least trios to calibrate that? What should there be a bulk sequencing also of DNA that's in addition to the single cell? And then can we understand fixation of mutations and sort of when do cells if there's a chromosomal event, when does the cells then apoptose or versus fixate the mutation, and what stages leads to trajectories of harm? What do you think on that? Some big agendas.
Can I start answering the question about parental DNAs? I know certainly with a lot of the prenatal tissues and samples that we received, it's not possible at all to have parental DNA, and it's actually forbidden because it's explicitly stated in the consent form that there is no way we can trace it back to the parents. And that's fine, because if we're talking about de novo mutations, somatic mutations, then we can do some trajectory analysis even within the sample itself, right? So it would be possible to, make some sort of pseudo-time analyses in theory and say, at what point during gestation might this mutation have occurred, given what the progeny that are mutated are. So that's one thing, but you made a nice long question. So Stephane, maybe if you want to comment on other parts of it, I'd be grateful.
I think I agree with you on your comment.
Okay. What else was there? No, I'm sorry, Bruce, it was a long question. I don't remember all of it.
Well, and understanding mutant effects in time and space, and then complex allele interaction effects. There's so many agendas.
I think the important point here that seemed to be a general theme is that we have to keep track of all of this metadata, and it's really hard to know what we want initially, and sometimes you want to add it in later. It's hard to develop such a flexible kind of database, that's going to enable us to pull the data later on when we need to know it. And in particular, as one of our participants had brought up, go back to the DNA sample that was kept just in case, and to indeed test, at least in bulk, was that mutation present at a given time? How normal was that sample, or is it in fact a really great early example of, some sort of, somatic mutation that has an effect actually later in life, but we didn't see it because we took the sample too early.
Modeling this in organoids is going to be awesome too.
A very, very exciting frontier for sure.
All right, we better move on. Thank you so much. That was awesome. And there'll be...
Thanks to all the participants. We're really glad about that.
Yes.
Okay, let's see. It's Ali and Hiroki, please.
Good. So, hi everyone. On behalf of Hiro and myself, I would like to give summary of what we have been discussing yesterday. It was quite also fun and fruitful discussions around imaging and how to move next steps in combining imaging with molecular profiling. Most of you are much more familiar now, the technologies that we talk here around single-cell transcriptomics, as well as, of course, proteomics, which are technologies that allows us to do now unbiased molecular profiling at cell level. Clear that we have to also move to imaging at cell level. We need to be unbiased there, because most of these tissues that we analyze, they are cherry picked up from a part of tissue, which may be or may not be the most interesting part of the organ or organism. So, well, clearly, as we are an organism of interconnected systems from head to toe, we work as a system. We have to look at the whole system and identify what is anatomically interesting or pathological, then zoom in there and then perform our subsequent molecular profiling. So that should be ideally way, right? However, currently it's not the way. So what we do when we talk about spatial omics, that means we take a slice, 2D slice from somewhere, from a tissue, and analyze it using transcriptomics or proteomics. And while we have unbiased molecular profiling, we have a bias in image selection. If, for example, just we think that we are slicing mouse brain, usually this slice is about 1 in 1,000 of the whole volume. So clearly, we have to be very biased here to choose something and then do molecular profile. Of course, ideally, we should avoid that. First, we should image the whole organism at cell level, have anatomical, structural, and spatial location information, then perform the molecular profiling. That would be quite desired. But we have, of course, problems at the imaging site. If you think about cellular level imaging, it doesn't really exist at whole organ level. So if we look, imaging MRI, DTI, fMRI-like modalities, they can see whole organ, but they won't see the cells. We can do cell imaging with histology or EM, but then we are again looking at the piece. So the technologies that we, my lab, Hiro's lab, and other labs in the world developing so-called tissue clearing combines these two arms, that we can look at the gross anatomy at subcellular level in a time and cost efficient way. For example, we can even look at whole human organs, kidney at cell level and get those information. So then, of course, from here, the ideal thing would be to move to molecular profiling. So what is tissue clearing? So I want to give you a little bit more background here, as most of you probably less familiar with this field. So biological tissue opaque because refractive indexes are very different from between different components, protein, lipid, water, that scatters the light. So in tissue clearing technologies, we do some biochemical processes and then match the refractive index throughout tissues, then the light would not scatter, and it allows us to do laser scanning end-to-end at high resolution. So resulting tissues, actually, they are really transparent, literally transparent. For example, the whole organism, like whole mouse, becomes transparent. The air you can see is a glass.